Inside a dividing bacterial cell, the helicase enzyme splits the DNA double helix to expose the genetic code. This is an aggressively kinetic physical process. This schematic tracks the engine's speed. In E. coli, the helicase advances at 500 nucleotide pairs per second. Translating this forward velocity into rotational physics, the unbroken DNA duplex ahead of the fork is forced to rotate at nearly 3,000 revolutions per minute. This immense torsional strain creates a severe mechanical bottleneck. The twisting force accumulates to the point where it will stall the advancing DNA polymerase or shatter the entire chromosome. DNA replication poses an extreme biophysical crisis. Copying the genetic code requires continuous mechanical management to prevent the system from tearing itself apart. The severity of this crisis is a direct result of macro-level genomic structure. Bacterial genomes exist as covalently closed circles. The physical rules governing any closed loop are rigid. If you force a local unwinding of the strands at one specific site, the loop must compensate by overwinding somewhere else. Eukaryotic chromosomes are linear, but their massive size and periodic anchoring to protein scaffolding create isolated topological domains that function like closed loops. Because the ends are fixed and cannot freely spin to relieve the pressure, the 3,000 RPM torque from the replication fork has nowhere to dissipate. It compounds instantly as severe positive supercoiling. Locked ends mean that simple unwinding is physically impossible without breaking the DNA. The structural strain of replication becomes a lethal trap that biochemistry alone cannot solve. To understand how the cell survives this, we must examine the geometry of relaxed DNA in its baseline state. A relaxed double helix prefers a very specific microstructure, exactly 10.5 base pairs per helical turn. In any closed loop of DNA, the number of times one strand winds around its partner is known as the linking number. This is a fixed mathematical property that can only be altered if a covalent bond is severed. This diagram illustrates what happens under mechanical stress. When the linking number is mathematically forced to change, for example, overwound by an increment of +2, the helix must physically contort its 3D shape to preserve its preferred 10.5 base pairs per turn. The visual splits here to show the two geometric manifestations of this contortion. On the left, it twists upon itself like an old telephone cord, creating plectonemic supercoils. On the right, it wraps around a central axis to form spring-like solenoidal supercoils. Plectonemic supercoils are the only stable form found in naked DNA. Solenoidal supercoils, however, dictate the specific geometry of DNA when it wraps around biological nucleosomes. Supercoiling is never a random tangle. It is the strict mathematical reaction of a fixed linking number yielding to torsional stress. To manage this stress, cellular systems deploy topoisomerases. These are highly specialized mechanical enzymes designed to intentionally break the rules of topology. Type 1 topoisomerases relieve strain by introducing a transient break into exactly one DNA strand. This severs the topological lock, allowing the broken strand to swivel around the intact strand and bleed off the accumulated torque. Topoisomerase 1 executes this cleavage and religation without consuming a single molecule of ATP. It achieves this efficiency by temporarily storing the energy of the severed phosphodiester bond directly within a phosphotyrosine linkage inside the enzyme's active site.
Topoisomerase 1 acts as an elegant, energy-neutral release valve, constantly resolving baseline torsional strain across the genome. But the baseline valve is not enough for the replication fork. The 3,000 RPM positive supercoiling forced ahead of the helicase requires a far more aggressive, energy-intensive intervention. This is the domain of DNA gyrase, a type 2 topoisomerase engineered to manage extreme positive supercoiling. This schematic animation shows the gyrase mechanism. It binds a crossed DNA duplex and uses the energy of ATP to cleanly sever both strands. It then physically forces the intact duplex straight through the newly created gap before resealing the break. By passing one duplex through another, gyrase mathematically subtracts from the linking number by exactly 2. This instantly converts lethal positive supercoils into negative supercoils, clearing the path ahead of the advancing fork. Because this heavy-duty mechanism is unique and essential, it is a primary target in clinical pharmacology. 4 quinolone antibiotics specifically hunt bacterial DNA gyrase.
These drugs inhibit the final resealing step of the reaction. They freeze the enzyme mid-cleavage, filling the bacterial chromosome with fatal, unrepairable double-strand breaks. Without the ATP-driven double-strand breaking power of gyrase, the high-speed replisome would instantly paralyze itself with its own torque. Zooming out to the end of the E. coli replication cycle, we look at the macrostructure of the circular chromosome. As the two opposing replication forks converge, the physical danger shifts. Torsional strain gives way to physical entanglement. This diagram illustrates the organization of replication termination. The advancing replication forks are corralled into specific genetic sequences bounded by the TUS-TER complex, which functions as a strict directional trap. Because the parent genome began as a covalently closed circle, completing the process produces a geometric artifact. The two newly synthesized daughter chromosomes emerge physically interlinked. This interlocked geometric state is known as a catenane. The two massive DNA rings are trapped together like links and chainmail. To resolve this, the cell calls on topoisomerase 4. This specific type 2 enzyme executes a final ATP-dependent double-strand pass-through to unlink the two rings. Replication cannot conclude until this final topological knot is resolved. Only after topo4 unlinks the catenanes can the separate chromosomes successfully segregate into daughter cells. Researchers exploit these highly predictable enzyme mechanics to map the invisible topology of the genome directly in the laboratory. By incubating complex DNA protein structures with specific topoisomerases, scientists can force the removal of supercoils, locking the native topological state into a measurable form. This figure reveals the outcome of one such assay, testing cohesin and condensin. By incubating these structurally similar proteins with topoisomerase 2, they leave distinctly different geometric signatures on the gel. Condensin interaction forces the DNA into a trefoil knot, while cohesin generates a series of interlinked catenanes. The physical assay proves they execute mechanically distinct roles, even though they share similar structures. Topoisomerases serve as the primary mechanical regulators of the genome. Their precise control of the linking number demonstrates that in molecular biology, three-dimensional topology dictates function as much as the genetic sequence.